This is a first-in-human trial looking at a new antibody-drug conjugate for melanoma. So it's an antibody that targets ETBR, or endothelin B receptor, and targeting the endothelin B receptor by itself with an antibody is not predicted to cause anti-cancer benefit, or it's not going to cause the cancer cells to die. But the fact that this is preferentially expressed on over half the patients with melanoma, it creates a target potentially that we could use with an antibody-drug conjugate that carries chemo on the back of it, that you could then infuse this antibody-drug conjugate into the blood. The way these antibody-drug conjugates are developed are that the tips of the antibody, they're kind of Y-shaped molecules, are coded specifically for, in this case, ETBR, for expression of that, which should be preferentially expressed only on melanoma cells, and that the antibody-drug conjugate would travel through the blood. It has chemo on the back of it. So the idea of these antibody-drug conjugates are that you're attaching a very potent chemotherapy that normally couldn't be given intravenously, and these are actually very old drugs that have been around for 25, 30 years, but that are too toxic when they're given regularly as an IV infusion. But if you tack it to the back of an antibody, they remain inactive. And the idea is that it'll travel through the blood looking specifically for the receptor, carrying the inactive chemo, and then once it binds to the receptor, it has to get internalized. And once it gets internalized, that complex will be broken down, and that's part of the biology of how the linker releases the chemo, and then once it's released off the antibody inside the cell, it releases the active chemotherapy directly inside the cancer cell. When you're thinking about developing antibody-drug conjugates, one of the most important parts of that is to develop a companion diagnostic so you can see or predict what patients are likely to have expression of the target that you're trying to deliver the drug to. And so in this case, what we were trying to do is look at patients' tumor specimens and stain them for ETBR and see if that would eventually correlate with the patients that were getting benefit. So the hypothesis would be that if you had a high expression of ETBR in your cancer, then we could predict that you would be more likely, because that's what the drug's targeting, and delivering more chemotherapy per se to those cells. This is an early clinical trial. We treated 28 patients across multiple dose levels, and we did on 24 of the patients, we have what we would call radiographic responses, where you have scans before going on study and scans at least at the first tumor assessment, which is 6 weeks. And we also have IHC data for the companion diagnostic on that. And there's a trend that if you are someone whose tumor expresses ETBR, overexpresses or has a higher rate of expression of ETBR, that you're the one, that those are the patients that are the ones that are most likely to gain the clinical benefit. But it's early, the numbers are small. The study did do an expansion of 24 extra melanoma patients, and we need to make sure that that is correct. When we give the antibody-drug conjugate, we think that the primary toxicities are related to the chemo on the back of it, that some of the chemo eventually gets out into the bloodstream. Either it came off before it got into the cancer cell and became active, or once it destroys the cancer cell, it releases some of the active chemo into the bloodstream. And so there are inherent toxicities to these. The most common issue that we deal with in the clinic is fatigue. In this trial, was always grade 1, 2. We did not have any grade 3, 4 fatigue. But fatigue, I think, is an issue for patients. And that being said, we've had patients on for over 2 years at this point. So it seems that there are some patients that it is a very good drug and it's tolerable. Other toxicities include hair loss, which could be due to the way the drug targets the skin or melanoma, but also because the parent chemo causes hair loss, so we don't really know for sure. And then other GI side effects like diarrhea and nausea can happen. And then, particularly for patients that are on for multiple cycles, neuropathy becomes an issue. So that's numbness and tingling in the fingers and toes. And that's mainly due to the parent or to the payload, which is the chemo on the back of it. And that's a known toxicity of those class of drugs. So over time, I think about 30% of our patients developed peripheral neuropathy. Most of the time, though, if you looked at all the side effects, almost everything is grade 1, 2. The grade 3 changes that we've seen were neuropathy or neutropenia, but that can tend to be manageable in the clinic and doesn't change dosing. One of the interesting parts of the study was that it included patients with cutaneous melanoma, which are the most common, but also allowed patients that had mucosal or ocular melanoma. There were 3 patients with mucosal melanoma and 8 patients with ocular melanoma. And we had clinical benefit, which would be either a partial response or someone that stayed on study for 6 months and maybe had minor radiographic changes, but not enough to meet the criteria of partial response, but was able to stay on the treatment and hold their tumor at bay. And we had evidence of that in both those subtypes of melanoma, and that's kind of an unmet need right now in oncology for both mucosal melanoma and ocular melanoma. I think with the results from the escalation look very encouraging. That we've had clinical, I think what we'd say it would be proof of concept that we showed that we can give the drug safely. When we give the drug, we can measure levels of it in the body, and it's behaving the way we expect it to, and people can tolerate it and stay on it very long. And then we also had what would be most important is that patients that were doing well from it and their tumors were either shrinking or stable and could stay on it a long time. But it was only 28 patients, and we treated patients across multiple dose levels. And so now that we have the recommended dose, there's an expansion of 24 patients to try to confirm that the tolerability is as good as what we saw initially, and also that we're seeing the clinical activity. And then based on that, we'll make a decision whether to go further in all melanoma or cutaneous melanoma or ocular or mucosal melanoma.